In this video, we'll discuss about megaloblastic anemia. Anemia is due to deficiency of vitamin B12 and folic acid. Two things, either vitamin B12, either folate, any one of the two can lead to anemia. In a person is known as megaloblastic anemia. Why? Why vitamin B12 and folic acid can lead to anemia? Iron deficiency anemia means because of that iron defect. Iron required for heme synthesis. Okay, hemoglobin. Heme is made up of iron, so it leads to iron deficiency anemia. Here, not required for that formation of heme. They are required for formation of DNA. So here vitamin B12 and folic acid is required for formation of DNA. DNA make nucleus of the cell. So nucleus for synthesis of DNA we required vitamin B12 and folate. RBC does not required, but their precursor required. Okay, precursor required that nucleus. RBC formed in bone marrow. Okay, this one is the bone marrow. We have multipotent hematopoietic stem cells. In bone marrow, we have multipotent hematopoietic stem cells. It have nucleus. Then myeloid progenitor. It also having nucleus. Then erythroblast, it also having nucleus. Then basophilic erythroblast, it also having nucleus. Then polychromatic erythroblast, it also having nucleus. Then orthochromatic erythroblast, here loss of nucleus, removal of orbit. Okay, N nucleation, here nucleus is absent. After that no nucleus, so reticulocytes and RBC does not have nucleus. Nucleus is presented. Okay, these reticulocytes and RBC don't have nucleus, but that precursor, this myeloid progenitor, these precursors having nucleus. DNA is made up of 4 nucleotides: adenine, guanine, cytosine and thymidine. Thymidine formed from uracil. So uracil get converted into thymidine. This conversion, that uracil is converted into thymidine. This conversion we required vitamin B12 and folic acid. Imagine a person does not take vitamin B12 and folic acid in the diet, body stored also deficient. So uracil is not converted into thymidine. So remaining 3 things, adenine, guanine, cytosine are present, but thymidine is absent. This thymidine absent, so DNA is not formed. The DNA double helix replication not takes place. DNA double helix replication not formed means nucleus is not formed. So DNA not is there. All the steps is affected. That process not divided. So cytoplasm will move to next stages. So cytoplasm here will move to next stages, but nucleus is not moving. So that reason only that cytoplasm is not moving next stage, but here after that nucleus is not moving. Cytoplasm will move to the next stage. It will go to pronormoplast, then it will go to early normoplast, then it will move to intermediate normoplast, then last. Okay, after that cytoplasm will move to next stage, that is, it is known as nucleus lagging behind cytoplasm, or we can say cytoplasm leading ahead. Usually both should go parallel. Here nucleus and cytoplasm parallelly it will move, moving, but due to deficient nucleus not jumping, cytoplasm only moving. So here myeloid progenitor to cytoplasm will move to the next stage. So early progenitor, intermediate progenitor, and last, okay, progenitor. Here cytoplasm will jump to next stage, nucleus lagging and cytoplasm leading stage. So this one name is called as nucleus cytoplasm asynchronous. Normally there are synchronous, now asynchronous. This is the hallmark feature of megaloblastic anemia. Both are moving next stage, but because of that vitamin B12 and folic acid, that nucleus is not moving, only cytoplasm only moving. So it is called as nucleus cytoplasm asynchronous. What is megaloblastic anemia? Why we call it megaloblastic anemia? Here problem with the nucleus, not in cytoplasm. So cytoplasm more abundant. So it enter end stage. RBC or bigger in size. Precursor also abundant. Nucleus absent, but cytoplasm is abundant. RBC and precursor of RBC bigger. So it is called as megaloblastic anemia. So, megaloblastic anemia, here problem with the nucleus, not in cytoplasm. Cytoplasm more abundant. So, it enter end stage, RBC or bigger in size. Precursor also abundant, nucleus absent, but cytoplasm is abundant. RBC and precursor of RBC bigger. So, it is called as megaloblastic anemia. RBC and precursor of RBC bigger in size, so it have that name megaloblastic anemia. And that normally precursor are known as normoplast. Precursor are known as normoplast. They are early normoplast, intermediate normoplast, and late normoplast. All are large in size, so it is called as megaloblast. RBC are known as erythrocytes. RBC is also bigger in size, so we will call it macrocyte. Macrocytes. Okay. So inside bone marrow, we can see megaloblast, and inside peripheral smear we have macrocytes. So it is due to that nucleus cytoplasm asynchronicity. So here nucleus lagging behind and cytoplasm leading ahead. So actually both are parallel. Here not parallel. One is leading and one is lagging, not parallel. So nucleus lagging behind, cytoplasm leading ahead. Megaloblastic anemia because of nuclear defect. It is due to vitamin B12 and folate deficiency. Megaloblastic anemia or disorder caused by impaired DNA synthesis. Vitamin B12 and folic acid are required for DNA synthesis. Now, vitamin B12 and folic acid absorption, how it will absorb. Vitamin B12 presented in diet. All dietary and non-veg food material bind with binding protein. So this one is the dotted one is vitamin B12. It will bind with the binding protein. That V-shape one is called binding protein. Vitamin B12 bind with the binding protein. After eating, it will reach to our salivary gland of human. That salivary gland will secrete a protein name is called as hepatocorrin. So it will secrete a protein name is called as hepatocorrin. So it also enter to stomach. Here not binding, this hepatocorrin not binding, separate way also it also enter to our stomach. Stomach will secreted HCl. So it will bind with vitamin B12. This HCl will bind with vitamin B12. Now it will bind. Okay, now it will bind. Now only that hepatocorrin and vitamin B12 will bind. Now it will go to our intestine. In the stomach there are cell is presented. Okay, here cell is presented, parental cell. That cell name is called as parental cell. Will secrete intrinsic factor. In intestine we have alkaline medium, the bond. So here that bond is broken. Vitamin B12 is separated from that hepatocorrin. Now it will bind with intrinsic factor. Now only this vitamin B, here only after HCl only that hepatocorrin and vitamin B12 will bind. When it will reach to here intestine, this parental cell will secrete intrinsic factor. After that it will separated that protein hepatocorrin as well as vitamin B12. Now this vitamin B12 bind with intrinsic protein. It will finally reach to our jejunum. Finally jejunum will get absorbed. So intrinsic factor is important. Without intrinsic factor vitamin B12 cannot absorbed. It causing anemia name is called as pernicious anemia. Pernicious anemia, low RBC when body cannot be absorbed enough vitamin B12. Okay, our body cannot absorb enough amount of vitamin B12. It causing anemia name is called as pernicious anemia. What is the problem? Problem not vitamin B12. Diet we have vitamin B12. Here the problem is parental cell. Okay, here that problem is parental cell or death. It is due to autoimmune disorder or antibody killing parental cell. Okay, they do not absorb vitamin B12. This type of anemia name is called as pernicious anemia. Here we will take through dietary source we will take vitamin B12, but here that parental cell or death because of autoimmunity or antibodies are killing parental cell. So our body cannot absorb vitamin B12. This anemia name is called as pernicious anemia. It is a special type of anemia. Now etiology and pathogenesis, how vitamin B12 and folic acid can leads to anemia. Vitamin B12 deficiency, whether decrease intake, inadequate intake, or vegetarian. Vitamin B12 high amount presented in dietary and non-veg source. After that impaired absorption in taking diet not absorbed in intestine, whether because of intrinsic factor deficiency, because of pernicious anemia, or gastrectomy. No stomach due to cancer, gastrectomy is done, or intestinal diseases, Crohn's disease, like that, or bacterial overgrowth. What are the reason? Our body cannot absorb because of intrinsic factor deficiency, or intestinal disease, or bacteria overgrowth. Otherwise, increased requirement because of pregnancy, hyperthyroidism, or cancer. So these are the period our body required high amount of vitamin B12. In diet, it is going to mouth, esophagus, stomach. The stomach, it will bind with intrinsic factors secreted from parental cell of stomach, and in combination going to that intestine. Vitamin B12 with intrinsic factor, it will be absorbed from that intestine. What are the reasons for that deficiency? Diet may be deficient, pure vegetarian, or old age, or loss of appetite, or poverty. Anything can be there. Then this parental cell absent in stomach, or intestine not absorbed, or intestine having that overload bacteria. So these are the four important reasons for vitamin B12 deficiency. Next, folic acid deficiency. It is presented in green leafy vegetables. What are the etiology? Decreased intake, inadequate diet, or alcoholism, or infancy. Then impaired absorption, malabsorption, or intestinal disease. Then increased requirement. Then these are the other factors. These are the four reasons for folic acid deficiency. Vitamin B12 and folic acid is required for DNA synthesis. DNA having four things: adenine, guanine, cytosine, and thymine. How the thymine formed? Thymine is formed by conversion of uracil. Uracil required vitamin B12 and folic acid. Conversion of thymine. For this step, we required both vitamin B12 and folic acid. See this diagram given in Robin. Deoxyuriditate monophosphate is converted into deoxytrimonophosphate. Then this deoxythymine monophosphate is required for DNA synthesis. And in this one, this methylene tetrahydrofolate is reduced into dihydrofolate. This process is required vitamin B12. So THF is converted into THF. Okay, dihydrofolate is converted into trihydrofolate. That time we required vitamin B12. So in these both molecules are required vitamin B12 and folic acid. Any one deficient or both of them deficient, it leads to deficiency. Thymidine. So inadequate DNA synthesis, defective nuclear maturation. So cytoplasmic maturation in advance. Okay, so here maturation of nucleus lag behind to that of cytoplasm. Nuclear or cytoplasmic asynchrony. So this one is that normoplast, erythroplast. This one early normoplast, intermediate normoplast, late normoplast, reticulocytes, and RBC. Here all the things this we have that nucleus. In this fourth step, here we have nucleus. Here exudate, nucleus will exudate. Here only cytoplasm is presented, nucleus is absent. cytoplasm go next stage, but nucleus remain in previous stage. All the cell cytoplasm abundant. Nucleus or stringage. Nucleus or stringage. So all of them are bigger in size. So it it called a megaloblast. Say for example, in bone marrow. In this bone marrow, pro-normoplast, early normoplast, intermediate and late normoplast, then it converted into RBC. All are these all are normoplast. It entered to blood. All are bigger in size. So it have that name. All these have that name megaloblast. When it will reach to blood, that time these erythrocytes in blood, it is known as megalocytes. So, megaloblast in bone marrow, megalocytes in blood. Uracil required vitamin B12 and folic acid converted into thiamine. Then it is required for DNA formation. So DNA formation required vitamin B12 and folic acid. Both absent or anyone absent, thiamine not formed. So thiamine not formed means DNA is not formed. So leads to anemia, megaloblastic anemia. Additional feature of vitamin B12. Vitamin B12 also involved in the conversion of methylmalonyl coenzyme to succinyl coenzyme A, which is required for formation of normal neural lipid. So that deficiency of vitamin B12 but not in folic acid result in neurological future. What is neurological future? Loss of sensation, tingling sensation, numbness, all the things. So summary, only folic acid deficiency leads to megaloblastic anemia. Megaloblastic anemia along with neuropathy will leads to vitamin B12 deficiency. This methylmalonyl coenzyme A converted into succinyl coenzyme A required for neuron. So it causes that neurological disorder, loss, sensation, tingling sensation and numbness. This video discussed about megaloblastic anemia. Bone marrow, megaloblast, blood megalocyte, future megaloblastic anemia. These are the morphological and functional abnormal cells. So megaloblastic anemia, morphologically and functionally abnormal cells.